In this part of the video, we are going to study about the concept of immunization. Now, how immunity develops and how do we immunize the person. So, let's talk about how immunity develops. So, first of all, let's talk about the primary immune response. Now, when a person is injected or is given a vaccine, then what happens? When a vaccine is given first time, the immune response which is received, that is called as primary immune response. Now you can see here, this is the time period and this is the concentration of antibody, that how much antibody is generated in the body. We know antibody is generated in response to the presence of antigen. So when first dose is given, this is initial exposure, there is no net production of antibody, but slowly there is production of antibodies. The first one to produce are immunoglobulin mu and immunoglobulin gamma, and then there is decline in its production. Now when there is second encounter with the antigen, or you can say when booster dose is given, the memory cells of these B cells, they are still present. They again form plasma cells and they again produce antibodies. Now this response is much higher as compared to the first response. So it is called as a booster response. That is why we can now understand that why booster doses are given, because whenever again that antigen enters into the body, the immune response is more as compared to the first encounter. Now vaccination is based on this thing, that when there is subsequent addition of the vaccine or introduction of the vaccine in the body, then there is increase in the immune response due to the presence of antigen. Now first of all, let's consider what is vaccination. It is the process of introduction of vaccine in the body. When vaccine is introduced in the body, the process of introduction of vaccine, that is called as vaccination. Now vaccine is that chemical preparation which contains the antigen that triggers the immune response. So when the vaccine is introduced in the body, the process of that is called as vaccination. Now what is a vaccine? Vaccine is preparation or extract from the dead germs of the disease. Like if we are going to talk about polio virus, then the virus is introduced in such a form that it is not able to cause the disease, but it triggers the immune response to produce the antibodies for the same. So either the germs are dead or they are attenuated. Attenuated means they are weakened. They are in such a medium that they are not able to cause the disease. But that when these are introduced in healthy individuals, that leads to development of the antibodies. Then next concept is of immunization. Now there are two concepts. One was vaccination. Vaccination was the process of introducing the vaccine in body. Now what is immunization? Now when the vaccines are introduced in the body, our body produces antibodies against the vaccine, because vaccine is nothing but it is a sort of antigen which is introduced. Now when the antibodies are produced, this generates a sort of immunity, and this we already studied is a kind of artificial active immunity. It is active immunity because it is generated in our body. And why it is called artificial? Because it is due to the response of the vaccine which is given to the body. So that is leading to immunity development. So it is called as immunization. Now when the subsequent doses, more and more doses of vaccines are given after certain time period, the response of B and T cells, which are specific immunity, is more. It leads to more amount of production of antibodies and cells, lymphocytes. They are producing large amounts. So that is why, to increase the immunity process or immunization, booster doses they are given so that we can protect ourselves from the disease. Now regarding the types of vaccine, the question arises, what are the different types of vaccines over here? Now first of all, first type is inactivated whole agent vaccine. Now here inactivated, it means microbes are killed. Whole agent means total microbe is inserted in the body. Not a part of the microbe, but whole of the microbe is inserted into the body through the vaccine. Examples are vaccines against rabies, influenza, Salk vaccine, pneumonia, cholera. These are different whole agent vaccines which are inactivated. That in this case, the dead germs of these diseases are introduced in the body. Then are attenuated whole agent vaccines. In this case, the vaccines use living organisms, but they are attenuated. Attenuated means they are weakened. They cannot cause the disease, but they are introduced in the body. Example, Sabine polio vaccine, MMR vaccine. These are the examples of attenuated whole agent vaccines. Now, in these two cases, there was whole agent vaccine. Whole agent means total complete body of the microbe that is inserted in the body. But then second case is subunit vaccine. Here whole of the microbe is not used, but only its antigenic fragment, that is a part of the microbe which leads to the production of antibodies, that is antigen part, that is only introduced in the body, and its just purpose is to stimulate the immune response. And some of the vaccines, they are produced by genetic engineering. We will be studying about genetic engineering later on. In this case, we are able to use a particular fragment of the DNA which is required by us. So here other organisms, they are modified to produce the antigen fraction of the germs, and these are called as recombinant vaccine. Actually what happens in genetic engineering, the required part of DNA which is producing a particular kind of protein required by our body, that part of the DNA is introduced in some other organism, and the other organism in which that DNA is introduced is now able to produce that very amount of chemical which was required by us. So here the antigen, we need antigen of one microbe, that part of the DNA which is producing that antigen is introduced in some other microbe which is not a disease causing microbe, and that microbe is producing that antigen also, and that are introduced in the body. Example, vaccine which is against hepatitis B, it contains a genetically modified yeast. Now yeast is otherwise a fungus, it is not causing a disease, it is not a virus, but here the gene of the virus is introduced in it which produces the viral protein coat. Otherwise it is not going to produce viral protein of the virus, but it is producing it because it is genetically modified. Then are the toxoids. Toxoids are actually chemically modified toxins. Sometimes the pathogen, they produce some kind of toxin. That toxin is chemically modified and it is not now pathogenic. It cannot cause the disease, but it is still antigenic, that it can trigger the body to produce the antibodies or the immune response. Such vaccines are called as toxoids because they have the they use the antigenic properties of the toxins of the microbe. Examples of these are vaccines for tetanus, diphtheria. These are the examples of toxoids. Now next are the conjugated vaccines. Conjugated vaccines, they are the vaccines which are produced by the their polysaccharides, they are combined with the proteins. Now this is due to the poor immune response in children. When children, they do not respond properly to these vaccines, they are not able to develop immunity or they are very weak, then in such cases the combined vaccines or conjugated vaccines, they are given. Example is for Haemophilus influenzae type B. Then are the nucleic acid vaccines. These are under trial. There are efforts being made to develop such vaccines. But here what will be going to happen? The DNA fragments, they will be injected to generate some kind of proteins which will stimulate the immune response. So here it is directed towards the introduction of DNA. Then is classification of another type of classification of vaccine on the basis of the time period. Now first is first generation vaccine. These were produced by the conventional methods, these traditional methods like introduction of the germs directly, or a part of germs directly into the body. Examples are smallpox vaccine, salk polio vaccine. Then second generation vaccine, they are little advanced. Here we use the technique of genetic engineering, like we just studied. We are modifying a yeast to produce the viral protein. So it is a sort of genetic engineering. So these vaccines are genetic second generation vaccines. Examples is vaccines for hepatitis B, herpes, influenza. Then third generation vaccine, these are synthetic vaccines, and these are still under trial, where we are still trying that we are not going to use the microbes or their parts, but actually we are going to synthesize some chemicals which are synthetic, and they can be used to trigger the immune response. So there are trials to produce vaccines for AIDS, malaria, hepatitis C, leprosy. So these are different kind of vaccines which we studied today. Today we studied the concept of immunization, vaccination, and the types of vaccines. Now in the next part of the video series, we'll be studying about the other concepts related to immunity, like we'll be studying what are antigens, what are antibodies, how they interact, and so on. Thank you.